Hey Bio30s, I'm here today to extinguish any doubts that you might have about this genetics unit. So let's get into it. We are going to finish up here talking about linked genes and chromosome mapping. So back to Thomas Morgan, he discovered another kind of interesting thing here related to chromosomes. Most of the time we think of chromosomes as, containing multiple genes, and then when we think of various genes in the body, they're associated on different chromosomes. So for example, eye color has nothing to do with hair color, and hair color has nothing to do with height. These are traits that are independent of one another, as Mendel said. However, linked genes would be genes that are found on the same chromosome. So genes that are found on the same chromosome and are often inherited together. So for example, on chromosome 4, we find the genes for red hair, freckles, and fair skin, which sometimes will be linked as a package because these traits will be all found on that same chromosome. So during meiosis 1, we need to remember that during meiosis 1, there's the process of crossing over. This is where genetic information can be swapped, right? So pieces of one chromosome can swap with another piece from a different chromosome. That being said, linked genes can become unlinked. So for example, genes can cross over. Oops, let's open this up here. So what I'm getting at is, let's say for example, we have gene A up here and gene E down here, okay? Now, normally those genes would be inherited together. They're found on the same chromosome. But what if crossing over occurred where this piece of the chromosome switched with a different piece? Now these two genes have been separated. So now one went over to the other one, and then that was replaced with a different allele. So we can unlink genes, number one, and number two, we notice that genes that are very, very closely located on a chromosome, so A and B are really close together, the chances of them splitting up are highly unlikely since they're so close, because let's say we break off this piece of the chromosome, right? A and B are still together. So the greater the crossing over frequency, or the greater the crossing over percentage, the farther apart genes are on a chromosome. So A and E are the most likely to cross over in this scenario, where A and B are the least likely to cross over because they are so close together. Now, as I said before, back to Thomas Morgan. Once again, he did the fruit fly experiments. He noticed that genes were found in the X chromosome. He was noticing that males were inheriting these genes more than females. He would also notice that, for example, if we have a dihybrid cross here, the wild body color with straight wings, both dominant traits, black body, curved wings, both recessive traits, he would expect all of the F1 offspring to be hybrids. This should be what we would get. However, if genes are linked and the recessives always end up together and crossing over occurs, what would happen is we might get some variations in our expected ratio. And these variations we call recombinants. So these are recombinations that occur during crossing over. So for example, if there are 300 offspring, you would expect all 300 to be hybrids. So they should all have the wild type body color and straight wings. However, only 282 of them had the wild type and straight wings. 9 of them were actually black with straight wings, so these two together, and 9 of them were wild with curved wings. So we can see that there's some crossing over occurring where we still have a dominant trait, but then a recessive trait as well for the other one. So seeing that crossing over occur on the chromosome. Now, what we would do then is to figure out the percentage of crossing over, or the recombinant frequency. We would simply take the number of recombinants divided by the total number of offspring, multiply it by 100 to get a percentage. So for example, if 18 out of the 300 were recombinants, that's a 6% crossing over frequency, or a 6% recombinant frequency. We can then determine, since it's 6%, those genes are very close together. So the chances of them actually splitting apart or unlinking are quite low. We can also determine that the 6% can be mapped onto a chromosome map. So we can actually map that 6% and figure out how far apart they are on a chromosome map. So when we do this, very, very simple, 1% will equal 1 map unit. So if there's 6% recombinant frequency, they are they're 6 map units apart. If they're 10% recombinant frequency or crossover frequency, then they are 10 map units apart. So how do we draw this chromosome map? Well, there's a few ways to go about it. Let's say A is in the middle of our chromosome map, okay? So here's our chromosome map here, right? A is going to be in the middle, so I'm going to put B and C on the outsides. Now, the distance from A to B is 12%. So that means A would be about there. So we're going to have 12 here. I like to always write the numbers in so we can check to make sure everything's correct. B to C is 7%. So 7 map units apart. And then A to C is then 5 map units apart. Now here's the thing we need to look at. Do our numbers add up? And as you can see, they do not, right? 12 plus 5 does not equal 7. That means A cannot be in the middle of this chromosome map. So it's a good thing to always check your numbers to make sure that your chromosome map makes sense. That map does not make sense. Let's try C in the middle. Let's put C in the middle. So that means A and B on the outsides. So A here, B here, A to B, 12 map units. We know this. A to C is going to be 5 map units, and then C to B is going to be 7 map units, and 7 plus 5 will equal 12. So the correct sequence is A, C, B, or B, C, A. Both would be correct. So A and B are farthest apart. They would have the highest chance of crossing over. A and C are the closest together. They would have the lowest chance of crossing over. And the order that they are on the chromosome would be ACB or BCA, depending on how you drew it out. So let's do another example here. Let's say we have genes DEF, right? We have 200 fertilized eggs, and these are the number of recombinants. So all we need to do is first determine the recombinant frequency, and then map our chromosome. So let's start here, D to F. We can start there. So we'll start D to E. Sorry, D to E. D to E, 22 recombinants. So we have 22 recombinants out of 200. All right. Next, we have D to F. So D to F, which is 7 recombinants. Then we have E to F, which is 15 recombinants. So E to F, which is 15 out of 200. Okay? So now we're going to take these, multiply them by 100 to get a percentage. All right. And I'll just grab my calculator to do so. So 22 divided by 200 multiplied by 100 gives us 11%. 11% recombinant frequency. 7 divided by 200 multiplied by 100 gets us 3.5% recombinant frequency. And then finally 15 divided by 200 multiplied by 100 gets us 7.5 recombinant frequency. Now, some of you can probably see what the map will look like, but let's draw it out just to make sure. Now, this is a trick I like to do. It doesn't always work, so you need to make sure the numbers add up. But I like to look at it and be like, okay, D and E are the farthest apart. I'm going to put them on opposite ends of my map. So I'm going to put D here. I'm going to put E here. And then just to keep track of everything, I'm going to know that they're 11 map units apart. Okay? D to F is going to be 3.5. And then E to F is going to be 7.5. So D to F, 3.5. And then E to F, 7.5, we know that 7.5 plus 3.5 equals 11, so everything adds up. So what is the sequence of this chromosome map? It could be D, F, E, or E, F, D. And then we can use these maps to once again determine the order of genes on this chromosome and the likelihood that they will unlink or stay linked together, which explains why some traits are always inherited as a package, such as red hair, freckles, and fair skin. So that's it for today's lesson. Thanks again for watching, and we'll chat soon.